Differential media, on the other hand, is media that helps tell the difference between different organisms depending upon the appearance of the colonies or the medium. So here's an example of a differential media you will be using in lab. Blood agar. Some organisms produce enzymes that partially break down the red blood cells and produce a green alpha hemolysis. Other organisms produce enzymes that completely break down the red blood cells so that you can see the yellow agar that the blood was mixed into. And then you have some organisms that can grow very happily on the blood agar, but they're not breaking down the blood cells. So we've been able to differentiate between these three different organisms based on whether they can break down the red blood cells and by how much. Oftentimes, we combine selective and differential media. It means we only have to streak our inoculant onto one media. McConkey is an example of a selective differential media that you will be using in lab. And we put in a dye that inhibits gram-negative bacteria, crystal violet to be specific, and then we put in just lactose so that we can tell those gram-positive organisms that ferment lactose and those that don't ferment lactose. How do we tell if the organism living on the agar is fermenting the lactose? Well, what's a byproduct of fermentation? Oftentimes it's an organic acid, which will lower the pH of the media. So we put a pH indicator in that will change colors. So here's an example of standard media, selective and differential media. So here we've taken two pure cultures, E. coli and Staphylococcus aureus, and we've streaked them onto standard media, non-selective differential media. E. coli is a gram-negative, Staph aureus is a gram-positive, and they're both growing very happily. When we streak the same cultures onto McConkey agar, the Staph aureus either doesn't grow or it has very little growth. The E. coli, on the other hand, very happily growing. Now, over here, we've streaked 2 gram-negative bacteria, E. coli and Salmonella, and you can see that there's a difference in the media by how the media behaves when they grow on it. So it's selective, selects against a gram-positive, and it's differential. We can tell the difference between these two organisms. So, thinking back to chapter 6, we know that some organisms cannot grow in the presence of oxygen, so we grow them in anaerobic media. Anaerobic media protects strict anaerobes from oxygen, and we can either do this by doing deep stabs, which would be to take a tube that has had agar poured into it, and we take an inoculating needle and stab it down deep into the agar. The organisms that end up down at the bottom are protected from oxygen. We can also use reducing media. Reducing media has molecules such as L-cytosine or sodium thioglycolate that reduce oxygen to water, thereby limiting the amount of free oxygen in the reducing media. We also can use anaerobe chambers or jars. Anaerobe chambers are chambers like this, and we have sleeves that you can put your arms through, but we've removed all of the oxygen from the atmosphere. Generally, we pump a combination of nitrogen, hydrogen, CO2, depending upon the needs of the anaerobe. Now, there's also anaerobe jars that you can put your plates in. They're much less expensive than the anaerobe chambers. And we have a catalyst that takes hydrogen that's released by these little packets, and the catalyst combines the hydrogen gas and the oxygen gas and makes water out of it. Also, CO2 is released because many organisms that are anaerobic need a higher concentration of CO2. And then we have an indicator to show when we've removed successfully the oxygen. Now, your very, very strict anaerobes don't generally survive the period when the oxygen is being removed. You have to grow them in the anaerobe chambers. Besides the different types of media that we've talked about, we also have what we call special culture techniques. Now, low oxygen culture, like what we talked about in the previous slide, is considered a special culture technique. And one of the ways that we can do this is to use a candle jar. Now, a candle jar is what we used to use before we came up with the technology of the anaerobe chamber. Basically, we would take a jar, put our media in it, put in a candle and light it. And the lit flame would use up all the oxygen in the atmosphere, or most of it. When the level of oxygen dropped to the level where there's no longer combustion available, the candle would go out. And part of the combustion process produced CO2. Candle jars are wonderful for growing organisms that need higher levels of CO2 and lower levels of oxygen. They're not so good for growing strict anaerobes. You got to use an anaerobe chamber for that. But the organisms that like the higher CO2, lower oxygen, we call capnophiles. Animal and cell culture are considered special culture techniques. You need special facilities for them. Then we have enrichment culture. Enrichment culture is basically selective media with oomph. We're not selecting against anything, we're promoting the growth of one thing. So if we have an organism that eats petroleum, we would enrich the culture media with petroleum to promote the growth of that organism. Whereas in the process, we would get rid of those organisms that don't particularly like to live in a whole bunch of petroleum. But petroleum in and of itself is not toxic like dyes. So it's not different, it's not selective, but it is promoting the growth of the organism that we want. Now that we've gotten our pure cultures, we want to preserve them. You don't want to have to go through all of that again and again. Refrigeration is the first method of preserving cultures. Refrigeration slows down the enzymes of non-psychrophiles. So if you slow down the enzymes, you have slowed down metabolism, you've slowed down the growth, but you haven't killed them. Refrigeration doesn't kill microbes. Then there's deep freezing. That's where you put them in a freezer that is at negative 80 degrees C or colder, and that stops the action of the enzymes altogether. But you have to be careful when you're doing deep freezing that you don't have crystal formation. Crystals can slice through cells just like little knives, and it'll kill your organisms, and you'll have fewer organisms when you go to wake them up, when you go to culture them and study them. And so it's better if you put in substances that act like antifreeze. Glycerol and DMSO are the two most common kinds. Lyophilization is the fancy-dancy term for freeze drying. We freeze them and then we sublime the water off. Then we don't have to keep them at low temperatures. You can keep lyophilized cultures at room temperature, so less expensive. That's it for this topic. Here are reminders of what I want you to learn from it. And once again, make sure you write down any questions that you may have so that we can discuss them in lecture.